Now we will discuss in this module a very high-tech analytical technique which is equally used by chemists, by molecular biologists, by analysts and physical chemists, you name it. That technique is called HPLC, which stands for High Performance Liquid Chromatography. Now, this is a form of liquid chromatography which is used to separate the compounds that are dissolved in solution. Thus, mind you, this technique is used only for those samples which are dissolved in solution, which are soluble in solution of some sort of another. And the instrument which we use, it consists of, number one, as in all chromatography methods, reservoir of mobile phase, then pump, then injector, which uses to inject the sample, and the heart of this is separation column, that is the column which separates the compound, and then of course the detectors which detect those compounds and send the signal to the computer. Principle is basically that the compounds are separated by injecting a sample mixture into the column. In the column, the sample mixture which we desire to analyze, that is injected, and then the different component in the mixture, they pass through the column and differentiate due to the differences in their partition behavior between the mobile phase and the stationary phase. Remember, in the chromatography, always there is a mobile phase and there is a stationary phase, and between these two, what happened, that whatever the differences between the partition coefficient between the different molecules, that it separates. The mobile phase must be degassed to eliminate the formation of air bubbles. The first thing, as we see in a HPLC system, you will see that there are reservoirs of different solvents. Here we see the solvent 1 and the solvent 2. Then there are degassed, remove the air and all that. Then the mixing vessel, and it goes to the pre-column where it is separated, and further through flow splitter it goes to a sample. There is a sample injector port from where the sample is injected, and under high pressure with a pump it goes through the analytical column, and the analytical column, there is a different signals are generated which are sent to the computer, and analytical data is achieved. Now, the 4 types of the liquid chromatography, that is the partition chromatography, adsorption or the liquid solid chromatography, ion exchange chromatography, and the size exclusion or the gel chromatography. These are the different types of the chromatography techniques which are frequently used in the analysis of the molecules. Now, the composition of the liquid chromatograph is, there is a system, it has got solvent, then there is a solvent delivery system, which is the pump, injector, sample column, detector, which are the array of diodes, and then there is a waste collector, of course, what passes over there, and the recorder, which gives or which records the data whatsoever. HPLC is used in chemistry, biochemistry, in molecular biology, and in all sort of modern researches. It is used for purifying the chemical compound, for developing processes for synthesizing the chemical compound if you have to develop a process. And then, of course, because this is a chromatographic technique, you can use it for isolating the natural product, for isolating the different metabolic products which are there. Now, in quality control, to ensure the purity of raw material and to control and improve the process yields is also important, and then quantify the assays for the final product and to evaluate the product stability and the monitor degradation, that how the product which is there, how much pure it is and how much it has been degraded. Now, it can also be used for analyzing the air and water pollutant, for monitoring material that may jeopardize the occupational safety or health. That is all analytical technique, and for monitoring pesticide level in the environment. So, in lot of branches of the science, this HPLC is used. Now, HPLC chromatograph injector, that is their advantage or their function is, as a matter of fact, to place the sample into the high pressure flow in as a narrow volume as possible, so that the sample enters the column in a homogeneous low volume plug, and then to minimize the spreading of the injected volume. These are the method. These are the function. The shortest possible length of tubing should be used from injector to the column. That is, from injector to the column. This should be the shortest. And what are the different factors which affect this system? That is the concentration, the matrices, solvent effect, and the sample effect which is there. Now, HPLC column, which is the heart of that, is one of the most important component of HPLC chromatograph. And HPLC column, the separation of column is achieved when those components pass through that column. Normally, columns are filled with silica gel because it is of a particular shape, surface, and properties, and pore structure help to get a good separation. This is a HPLC column, usually how it looks, and the evaluation of these parameters are efficiency, resolution, and then inertness, retention index, column bleed, and capacity factor. These are, as a matter of fact, the evaluation parameter for a column. It should be efficient, it should be capable of giving a reasonable resolution, and it should also be inert. That is, it shouldn't react with the compound or with the sample, and then its retention index. That is, it shouldn't take long time that it should, you inject the sample and it retains there and giving you result, but retention index should be considered. Column, how you have to bleed it, how the sample comes out of the column for the separation, and then the capacity factor, that what is the different capacity which this column can hold. So you see that as the different techniques they are developed, they, as a matter of fact, they work together, and the molecular biologists, the biologists, genetic engineers, and all that, they take advantage of these techniques to precisely understand the mechanism of molecular biology.